So welcome everyone. My name is Howard Rundle. I'm the Executive Vice President of the Society for the Study of Evolution. I'm pleased to be here today to introduce our speaker. First, I'll just mention that this is part of our Graduate Research Excellent Grant seminar series, in particular covering our R.C. Lewontin Early Awards, which are designed to assist students in the early stages of their PhD by enabling them to collect preliminary data or to enhance the scope of their project beyond current funding limits. If you're not aware of these awards, I encourage you to visit the SSE's webpage and find out about them because they're a great source of extra funding to help graduate students. And this year we're trying out this seminar series where current award winners are giving a weekly talk. So this week I'm pleased to, and I'll just mention that for the seminar we're following our standard meeting code of conduct, which requires that interactions are intended to foster the respectful exchange of ideas. And I think a link to the full code is being posted in the chat. And today's talk is being recorded, just so you know. So it's my pleasure to introduce Erica Vong. Erica is now a PhD student in the Rajakumar Lab at the University of Ottawa, but I first met her way back in summer of 2019 when she started in my lab as an undergraduate co-op student, I believe, if I remember correctly, although she cycled through various positions in my lab: co-op student, work study student, and eventually an honors student. She was one of those students that you just want to keep in your lab. Although it was sad to see her go in the end, it was terrific to see her go on to graduate work after her undergraduate. And I think she's going to tell us today about some of that work. So without any further ado, I'm going to turn it over to Erica to tell us about decoupling of dimorphic scaling patterns in ants. Take it away.
Thank you so much, Howard, for the kind introduction. I want to first thank the SSC and the Graduate Student Advisory Committee for organizing and hosting this GREG seminar series. And then lastly, thank you everyone for joining today. I'm really excited to tell you about some of the work I've been doing with ants. And as Howard mentioned, the title of my talk is The Evolutionary Decoupling of Dimorphic Scaling Patterns in Ants. And I will focus on some of the work for my thesis, but also some unexpected results that I found along the way. So just as a quick overview from left to right, I will give an ant development introduction, an allometry introduction, show you some of the results from my research and what I plan to do in the future. So largely, I am interested in the integration of ecology, evolution, and development. So how environmental factors interact with our genomes during development to produce phenotypic variation for evolution. And so ants in general are an amazing organism to study because they can be found on nearly every terrestrial landmass on Earth, making them an extremely ecologically successful species. They've evolved many interesting morphologies that you can kind of see in the background here, but they've also evolved many different strategies for their survival. So the two major hallmarks of ants that have likely facilitated their global radiation includes the evolution of cooperative societies and their highly sensitive development to environmental conditions, which results in developmental plasticity. So regarding their cooperation, ants are social insects and they live in a group called a colony. And then within this colony, there is cooperative brood care. So many ants will take care of the young. There are overlapping generations of adults, and so they all work together to retrieve food and take care of other colony tasks. And then ants are also highly sensitive to their environment as they are developing, which results in polyphenism in ants. So from the same genome, different environmental factors will result in different phenotypes. So to give an example, on our left, we have a queen ant and then a smaller worker ant. The queen is winged, is much larger, can live for decades, and is highly reproductive. The worker, on the other hand, is smaller, only lives for a few months, and is infertile. And then in some species, the worker caste has further diverged into soldiers and workers. So we have the soldier here and the smaller worker over here. the soldiers and the workers showcase tremendous amounts of morphological variation, from overall body size, this being a lot bigger, and this being so much smaller, to more specific traits, for instance, like its huge head compared to the worker. So these phenotypes that are morphologically, behaviorally, and physiologically different are organized into caste in ants. And so we have like the worker caste, the soldier caste, and then we have the queen. And so just to reiterate, from the same genome, key environmental factors will influence their development and result in these different castes. So how does this happen? So this is the ant caste determination pathway. So if we start at the top very briefly, males are haploid and females are diploid. Males are important for reproduction, but they usually die pretty soon after, unfortunately. Thus, the ant colony is comprised mainly of just all females. So once you go down the female trajectory a little bit, there are key environmental factors at during like key windows in their development, such as temperature and photoperiod, that are mediated by a key hormone called juvenile hormone. So when those conditions are met, then you have an increase in juvenile hormone, so JH+, and this will cause a developmental switch for queen-specific gene expression, and the individual will become a reproductive, long-lived queen. And then if those conditions are not met, then you don't have enough juvenile hormone, JH-, and the individual will go down the worker kind of destined pathway. But then again, in some species, the worker caste has been further elaborated. And so there's another key time period where environmental conditions such as nutrition and social interactions, and if those conditions again are met, then this will cause another developmental switch regulated by juvenile hormone again. And if they are met, then the individual will become soldier destined, and if they're not met, then worker destined. And just as kind of like a side note, in the literature, these are like the soldier caste is also referred to as major worker, and the worker caste is referred to as minor worker. But for this presentation, I'll just be using the worker soldier terminology to talk about them. So these workers and soldiers have, again, very different morphologies, like their head, size, and shape. And for instance, like the soldier has these huge mandibles that are nearly like the size of the small ant, the small worker ant. And so these morphologies play a part in determining task specialization in the colony and subsequently their behavior. So workers are smaller, and within the colony, they play more of a colony maintenance role. So for example, we have a worker that's taking care of brood by moving this clutch of eggs. And workers are also typically foragers, so they look for food and they explore more. Soldiers, on the other hand, have, again, like their huge mandibles that they use to bite and break down food. And they're also they're also defenders of the colony because they have these huge like bulldozer-like heads. And again, those huge mandibles. So to summarize, we have the soldier caste that has evolved these extreme morphologies, like their big-headed mandibles, which results in task specialization within the colony. However, something really interesting is that these extreme soldier morphologies are actually maladaptive in solitude. So William M. Wheeler found that if these soldiers fall on their backs, they actually remain in this position for hours or even 2 or 3 days, struggling at brief intervals and finally dying of hunger or fatigue, which is kind of drastic, but also really cool to read and learn about. So in solitude, they would have died, but in a social context, another ant can come along and help the soldier flip back onto its front and, you know, live and eat and all that kind of stuff. So the social aspect of ants has likely allowed for the evolution of these extreme morphologies by turning something maladaptive into something possible. So just the other day, I actually caught this phenomena on camera, and I've just kind of been showing everyone because like it's just kind of fun and cool to watch, including everyone that's tuned in for this presentation today. So in this video, you'll see a soldier ant on its back struggling because it can't flip back over. But you'll see that a little worker will come by and help it turn back onto its front, which is really cute because like these guys have like heads that are basically the size of or like 3 times the size of its body. And again, so like this is a maladaptive phenotype, but in a social context, it's possible for these elaborations to occur and to persist. Okay, so back to ants and development. So ants are holometabolous are a holometabolous species, meaning that they undergo complete metamorphosis and have distinct larval and adult forms. So the ant life cycle consists of 4 main stages. It's divided into the egg, the larval stage, the pupal stage, and then the adult ant that's just kind of like walking around. So ant eggs are like these tiny, smooth, jelly bean looking structures. And then after some time, they'll grow into these larvae. And it's at the larval stage where the most growth occurs. And then after a couple of moltings, the larvae will metamorphose. So that's this transition here into pupa. And this is a stage where they'll continue to further develop until they're ready to emerge as adults. Excuse me, sorry. So in whole metabolous insects, appendages develop from a imaginal disc. And imaginal discs are populations of cells that will pattern and grow to give rise to adult appendages. So in this illustration of imaginal disc, we have a Drosophila larvae, and the cell populations are colored and annotated with what they'll become after metamorphosis. So we have larvae, metamorphosis, and then adult. So for example, this leg disc over here, after metamorphosis and proper growth and all that, will become a leg. And so it works the same way in ants. There just isn't a pretty schematic like they have for Drosophila. And these discs are really important to study different appendages and how they develop, which is where my interests lie. So broadly, I am interested in the study of the evolution of morphology at the developmental level. So how do you pattern the size and shape of different appendages? So ants have a wide range of different traits that are incredibly interesting to study, including, their head shape and size, to their eyes and their antenna. However, the morphology that fascinates me the most is their mandibles. So mandibles are how these ants interact with their environment. You can see that the ant will use it to eat, to pick things up, also to scrape things off of their legs, kind of acting like a pair of hands, but not really. But from my intro, there's also variation in mandibles within species because of the worker-soldier caste, right? So I'm interested to see how you develop mandibles and how you create within species variation, but also I'm interested in a cross-species variation. So from this last page of ant heads, we see a massive amount of variation in teeth number, in length, and overall form. So from like these tusk-like mandibles that seem completely maladaptive again, but in a social context, it's possible. Normal mandibles that you probably would see in like bees, for instance, or these elongated ones that have, a bunch of teeth. So my overall objective is to investigate the underlying developmental basis of mandible diversity within and across species of ants. So to start off, I look at the scaling patterns within species of ants to quantify just the natural variation in mandibles before I do any developmental work. And so one way to study morphological scaling relationships in animals, which is also termed allometry, includes observing how that trait grows in relation to body size. So it's relative growth, basically. So one pattern that we see is when an individual grows larger, some trait of interest will grow proportionally to body size in a one-to-one manner. So what that looks like on this graph of body length to trait of interest would be like a slope, like a line with a slope of 1. And an example would be like the legs of this crab. growing proportionally as you get a larger crab. And then another pattern is when a trait of interest grows disproportionately larger with increases in body size. So we can look at the same crab and notice that the pincers are growing a lot larger than we would expect for its body size. So on a graph, it would be like a line with a slope greater than 1. And it's important to note that within an individual, different body parts are modular and can have their own scaling properties. So within one individual, the legs can grow proportionally, but other parts can grow disproportionately. And then a third pattern is where a trait of interest is disproportionately smaller with increases in body size. So you're going to see a weird picture coming up, but basically on the graph, you'll see a line with a slope less than 1. And I just like to note that this picture here, I guess like the little human cartoons are scaled or corrected for, I guess, like their body size to really emphasize how disproportionate the relative head size of humans is compared to the rest of its body as you develop. So these are all patterns that are observed and well studied in many organisms. And scaling in general has been a pattern of interest for a long time. So some of the most prominent examples includes the transformation grids, which you may be familiar with of Darcy Thompson, where he used a grid system to make shape changes to explain morphological evolution. But also Julian Huxley's book on problems of relative growth, where he explores allometry in biology and even has ants on the cover. So ant allometry has been of interest for a while because of this worker-soldier situation. And so this is an example of an allometry plot in ants. So imaged here, I have workers on the bottom row and soldiers on the top. Graphed here, I have scape length. So that's the first antennal segment here. So it comes out of their head, and then this is the rest of their antenna. And this is often used as a proxy for body size because it has been shown to grow proportionately to body. And it's a lot easier to measure because the ants are usually curled. And so it's easier just to measure that one thing that's coming out of their head. And then on the y-axis, we have head width. And so this is a trait that is often used to group the two different castes because the head is typically disproportionately larger in soldiers, as seen in colored in red here, compared to the workers in blue. So this is typically what is observed in ant species. There are traits that are, disproportionately larger in the soldier caste, like their head, and then traits that grow proportionately, like the scape, so that first antennal segment, and then traits that are disproportionately smaller that I didn't outline here. And so I have mandible allometry plots for this species, but it's not the highlight for today, so I'm just going to continue and talk to you about ants from the genus Phytolene. So this is a group of ants that have these really big-headed soldiers, and like with huge body sizes and these huge mandibles. So these ants are completely dimorphic, and they showcase, and this is like my focal species just because their mandibles are so distinct from one another. And so it would allow me to investigate their underlying developmental mechanisms, hopefully a little bit easier because it's more of a black and white situation when it comes to the mandible size and shape and other things like that. So I measured their scape, again, first antennal segment, and their body, and sorry, not their body width, their head width, just to recreate the graph that we saw in the previous slide, just to get an idea of what's happening in terms of their allometry. But what I find actually is something that really just didn't make sense. So I'm going to show you the same graph that I saw that we saw on the previous slide, but with data for Phytolea dentata. So on the x-axis, we have escape length, which again, first antennal segment, and then head width on the y-axis. So the pattern, if you look at the y-axis, the pattern of soldiers having larger heads and workers having smaller heads, this pattern stays true. However, the scape completely messes up what we would normally expect. So when we look at the x-axis, and we look at the points, we see that there is a complete overlap in morphological variation. So in this case, scape can no longer be used as a proxy for body size which is really confusing because we would have expected that the soldier would just have larger traits and maybe not, like, I guess we would expect it to be way larger, but in this case, they're completely overlapping. So for instance, if I were to pick a value of like 2.9 for their scape length, I wouldn't be able to tell whether it belonged to a worker or a soldier. So this suggests that scape length is decoupled from body size between castes. But looking at the data further, the distribution of variation within each caste is not random. We see an increase in scape length within each group from the linear regression. So in other words, we have like a small, a medium, and a large scape, and that corresponds in the soldiers as well. So for example, like the smallest worker will have the same size scape as the smallest soldier, and also the largest worker will have the same size scape as the largest soldier. So there is a little bit of a pattern here, which is really interesting because those ranges are like nearly the same for the workers and the soldiers. And this is a stark contrast from caste-specific traits like mandibles, where you can clearly see what belongs to a worker and soldier, whereas the antenna, you wouldn't be able to really tell. So let's see. So when I actually when I first collected this data, I was really confused because it hadn't looked like a graph that I'd seen in the literature at all. And so when I showed it to my supervisor, he was equally as confused and honestly really questioned if it was real. And so we did a couple of other things. Let's go. So what I did was I also measured other traits that have support for being proxies to body size. So here I have pronotum width, and on the x-axis and head width on the y. And so that's like this length here, just like across their thorax from the dorsal side. And then I have hind leg tibia on the x-axis in this bottom graph, head width on the y-axis. And so that was just like, the hind leg tibia is this measure right here. And so both of these traits follow what we would normally expect, that soldiers have larger traits than workers. And although we cannot say for sure that these traits are proxies for body size, because I just didn't test it, they at least follow the idea that soldiers have larger traits than workers. And so whether it be proportional, disproportional, disproportionately larger or smaller, they still there are still other properties of this ant that do follow regular scaling patterns that we would normally expect. And so in conclusion, something really funky is going on with the antenna. And so to investigate this further, we decided to tap into the amazing resource of a textbook called Fidoli in the New World. And this is written by Edward O. Wilson. And this textbook, and in this textbook, there are beautiful drawings of soldiers and workers and information on their natural history, ecological ranges, and morphological measurements, including their scapes. And so I have all this scape information on 626 different Fidoli species. And so I digitized this data with the goal of seeing if the escape pattern that is present in Fidoli dentata is also present in other species in this genus. And so what I do is for each species, I take their dimorphic scape ratio. So dimorphic scape ratio is the soldier scape length divided by the worker scape length. And if the ratio is greater than 1, it would mean that soldiers have a larger scape than workers, which is what we would expect. And then on a graph where we have ratio on the x-axis and number of species on the y, those values would fall, above 1. And then if the ratio is equal to 1, again, soldiers and workers would have the same scape length as we saw in Fidoli dentata. And then finally, if the ratio is less than 1, workers would have scapes that are longer than soldiers. And this would really be what we're not expecting. And so I took the dimorphic scape ratio for the 626 Fidoli species and created a frequency plot, much like this one that you see here. And what I find is that there is a normal distribution of the dimorphic scape ratio converging pretty close to 1, slightly above 1. So this suggests that on a macro scale across the genus Fidoli, the scape length is actually the same in soldiers and workers in many species. But that there are also many species where this isn't true. So I verify a few species using another amazing ant resource called antweb.org, which has images of pretty much all ants. So I find more species that have a dimorphic escape ratio of 1, highlighted here, like Phytoli hedgehoi, if I'm saying that right, and take some measurements just to verify, because I wasn't sure from the textbook what their sample size was because they didn't really outline that well. So I was just kind of double checking with the images that are publicly available to everyone. And so I took the escape length of the soldier and compared it to the worker. And if you kind of see, like, they're pretty much the same. But then I also checked the extremities to make sure that those are real as well. So if we go for a DSR that is greater than 1, we have Phytoli palula, where the soldier scapes are larger than the workers. So when you see the red line here, it extends past the scape, which ends around here. So that makes sense. But then I checked the other extremity, one that we really wouldn't expect, and see with Phytoli avia that there are soldier scapes that are actually smaller than worker scapes, which is really interesting. And I definitely want to look into that some more because it's kind of brain knocking a little bit. But anyway, so if you'll humor my Photoshop skills for a moment, I'd like to give you a decoupled dimorphic scaling pattern analogy. So if we return to a previous example where we have like a small crab and a big crab, if the decoupled dimorphic scaling pattern were true for the crab's pincers, we could have a small crab with a small pincer and a big crab with a big pincer. So that would be around this area here of this pattern. But we could also have a big crab with a big pincer and a small crab with a big pincer. So that's around this area of this pattern. And so I know this looks like ridiculous, but this is basically what the decoupled pattern is, saying that like these two different like morphological groups can have overlap in their traits. And so what is the functional significance of this morphological variation? So first, let's talk about scapes. So scapes are an elongated first antennal segment, and this is a synapomorphia ants. So other insects have shorter scapes, like for their first segment, and it's not elaborated the same way that it is in ants. So mechanically, it allows them greater range of motion to sense the chemical and olfactory world around them. And so since these are ants, these chemicals are kind of what language is to us, right? It's how they communicate. You can see that the ants are basically tasting the ground as they walk here. And then in this video, we see that the ants are touching their antenna to communicate with one another, whether it be about conspecific aspects or, sharing pheromones to tell other information. And so what is the functional significance of a decoupled scape pattern? So we're still not sure, but I've been reading more papers to understand how ants sense their environment in general. So one particular paper delved deeper into the behavioral strategies and patterns of antenna movement using an actively tracking ant. So what we have here is like a short video of an ant that's following a trail. And we see that it's basically like tasting the ground as it walks, specifically the most distal portion of the antenna, because that's where a lot of the chemoreceptors are located. But that the escape kind of just like, I guess, positions the rest of the antenna. And then in this video, the ant is also following a trail. So that's like that line there. But it's moving its body more in addition to its antenna. And so when we think about Phytoli and the soldiers, maybe having the same escape length as the workers allows them to follow trails better. So that they can navigate away and back to the nest, so they can follow the food trail to help break down things and just things like that. So in other words, maybe sharing a same escape length is just optimal for this species. Maybe. Still thinking about it. So on a micro scale in one species, I show the decoupling of a dimorphic scaling pattern, that there is no correlation of in escape length between the two castes, but that there is variation within each caste. So across the genus Phytoli, there are also many species that follow this pattern, so a dimorphic escape ratio of 1, but that there are also many species that diverge. And so this brings up a lot of questions about like what are the factors that are selecting and constraining its evolution, what role this trait plays in their natural history, and how this variation is generated throughout development. And so I'm going to work on answering those questions eventually, but what we decided to do next was to take a larger evolutionary view, because there are a lot of other instances of independently evolved worker-soldier castes across the ant family. So we wondered if this decoupled escape pattern was present in other species and just like was undetected. And so we targeted like, for instance, like the leafcutter ants, army ants that Robert and I were talking about just before, turtle ants, this is Carabera, I don't know what their common name is, and then the seed harvesters. And so here I have a phylogeny of ants, and then in red are, in red are annotations by myself to indicate species that have soldier worker castes for me to target. And so using the Lewontin grant from the SSE, I visited the Cornell University insect collection this summer in Ithaca, New York, to collect allometric data from key species highlighted in red on our left. So I'd like to just take a moment to thank the Moreau Lab at Cornell for hosting me, and specifically her graduate students, Chloe Jelly and Megan Barktel, for training me on using and working with museum specimens, how to use their microscope setup, booking, and just so much more. So here is a phylogeny that I have constructed just to show the relationship of the species I collected data for. And so for each species, there's a tiny little graph there. And it's the same graph that we've been seeing throughout this presentation. So head width versus escape length. And I'm just going to take a moment to go through a couple of the ones here that have worker-soldier castes. And so this data was collected from Cornell, from their museum's collection, but also just from some species that we have ourselves in the lab. So first, we'll start off at the bottom, which is the species most distant from Phaidole, dentata, which is at the very top. And we have Aceton burchelli, which is an army ant that have these soldiers with these tusk-like mandibles, which is really cool for my other interest. But anyway, we're going to see a lot of these graphs here, so I'm going to walk you through it again. So we have log head width, on the y-axis, and log escape length on the x. So soldier data points are colored in red, and worker data points are colored in blue. And so what I'm looking for is to see if the Phaidole decoupled escape pattern is present. So what we would see is like an overlap of their x-axis values for the red and blue points. So obviously we see that there is just regular scaling, that bigger individuals have bigger escapes than smaller individuals. So next we have Camponotus floridanus, which is a carpenter ant species. And again, we see regular scaling. And this is just a side note, like we have this species in our lab, so we were able to collect much larger data set than with museum collections. And then next we have Pogonomyrmex badius. So this is a seed harvester ant species, and again, just regular scaling. Next we have Acanthomermex thailandendis. So these guys have super huge heads, but again, their escapes follow regular scaling patterns because there is no overlap. Next we have Carabera diversa, so another species with huge heads and regular patterning. We're getting a little bit closer to Phaidole dentata on top. And we're looking at Atta mexicana. So this is a leafcutter ant species, and their soldiers have escapes that are bigger than workers, so nothing surprising. But then we get to Cephalotes varians. So this is a turtle ant species, and oh my goodness, there's overlap. So soldiers in this species have escapes that are similar sized to their tiny little workers, and we see pictures of these ants just on the side over here. So I was only able to get a few measurements, but I am working towards hopefully increasing the sample size for this just to get a clearer picture of what's happening allometrically in the species. And so with all of this information, we potentially found the origin of the decoupled escape pattern originally discovered in Phaidole, with the common ancestor being at of Phaidole and Cephalotes here. And so some next steps include taking a Phaidole specific approach to collect allometric level resolution of the escape using target species from this frequency plot that I got from the textbook. And so I plan on sampling from the extremes and also just like throughout the distribution, just to get a clear idea of what's happening in this genus. And then secondly, I would like to investigate the underlying developmental mechanisms that pattern the size and shape of antenna in ants, since first this hasn't been done, and also to understand the contribution of what genes are contributing to the elongation of the scape, since this is a synapomorphy of ants. And then lastly, I'd also like to try and understand what is contributing to the uncoupling of scape from body size. So what are some of the genetic factors that are playing a role in this? So now that I've told you about my detour, I'm going to talk a little bit about mandibles again. So this antennal work is truly unexpected, and it is truly an unexpected finding. Oh my God, sorry. And it has really snowballed into something bigger, but I still retain a lot of my original interests, and I'm still working towards understanding the development of mandibles. And so, like with antenna, I quantify the shape of mandibles for phydoid antenna to get an idea of what the wild type variation is. And so mandibles in a developmental context are really poorly studied in ants. So here I have a soldier mandible and then a worker mandible, and some morphometric measures that people take include the masticatory margin. So that's from the most apical tooth to the most basal tooth. The basal margin, which is the most basal tooth to the articulation here, and then the external margin, which is from the most apical tooth to the tip over here. And so for my needs, this unfortunately reduces the mandible to very simple shapes. And so I lose a lot of variation in, the teeth number, for instance, or the curvatures that are over here of the mandible. And so what I decide to do is to take a larger, I guess, more intensive shape analysis. So I perform geometric morphometrics on my mandibles. And so what this is a technique to study the scale and shape of structure. And so this technique allows me to place landmarks that have XY coordinates for many different spots, and to quantitatively analyze shape change between my two groups. So here I have like 9 homologous landmarks. So there's like 1, 2, 3, 4, 5, and then it goes to 9 here. And this corresponds between workers and soldiers. And so then these landmarks configurations are size normalized so that I can compare the shape of workers and soldier mandibles. And then from there, I can perform a principal component analysis to visualize the variation between these two groups. And so here I have plotted PC2 versus PC1. Soldiers are in red again, and workers are in blue. And we see like a very clear separation along the first principal component. And then the dark blue mandible like outline here represents the shape along this PC. So across PC1, we see that the most extreme over here is like a little bit wider, the teeth are a little bit larger, and then on the other end, it's a little bit more narrow and the teeth are a little thinner. And it corresponds with like soldiers and workers. And then for PC2, it's not as clear. There's a lot of overlap, but I didn't statistically analyze this, but I'm going to. So this is just wild type mandible shape. So my goal is to produce some RNA interference knockdown phenotypes of candidate genes that may pattern the shape of mandibles to observe what those genes are functionally responsible for during mandible morphogenesis. So I'm working towards that point. And so like I'll hopefully have a couple other groups that have like soldier RNA knockdown phenotypes to look at, I guess, like small shape changes, because those morphological differences can be really significant as well. So yeah, working towards this point, but so far I've just been describing mandible development. So here I'm going to show you pictures of wild type Phydoline minor workers throughout their pupal stage. And it is at their stage where mandible patterning occurs. So in general, we just see the progression of pupil development by the darkening of the eye here, and also the darkening of the exoskeleton. So regarding the mandibles, in the first 4 time points that I have imaged here, the mandibles have not fully patterned. We can see kind of like the general shape and size, but the teeth, for example, have not been fully carved out. But then for the last 4 time points, we see that the mandible teeth start to sclerize, so that's like the hardening of the exoskeleton. First at the teeth, and it starts to, I guess, like extend towards the base or to more the inner portion of the mandible. And so this pupal stage is really important for mandible patterning. This is a personal resource so that I can start to look at temporal and spatial gene expression using in situ hybridization. And this way I just have a wild type to compare to. And so the pupal stage is important for, yeah, mandible patterning. However, growth, for instance, so how large it, how long it extends and things like that, seems to have already been determined at this point. So what I'm going to show you next is the larval stage. So this is a dissection of an ant larva. And so these are all just like imaginal discs that I was kind of explaining in my introduction. And this is a Dapi stain of an ant, meaning that I'm staining like the nucleus of different cells, of all the cells, sorry. And so there's like 6 structures here. This one's a little floaty because it got moved, but these will all become legs. And what I'm trying to still do is to find the mandible imaginal disc because it hasn't been well annotated in the literature, and I'm still kind of just exploring the head region of the larvae to try and find it. I think it's this, but again, I'm not sure. So I'm still doing more molecular tests of different genes to try and get an idea. And then from there, I can observe how the mandible tissue grows, and also test what genes might be patterning their growth and proliferation, and just so on. And so with that, I'll just leave it at that. So thank you so much, everyone, for coming. I'd like to thank my supervisor and the undergraduate students that helped me collect a lot of this data, my lab in general, and then our funding agencies, and again, the SSE, for hosting this GREG seminar series. So with that, I'd be more than happy to answer any questions if there are any.
Thanks, Erica. That was great.
Thank you.
Um, I've got one question of my own. This this may not be directly in line with your interests if you're trying to understand the developmental basis of this uncoupling, but I'm curious, across the genus, if I recall, there was a broad array across species. There's quite a range of ratios of scape to, was it mandible? Or scape to body,
The
I
body.
guess. Yeah,
Yeah.
body. And
Sorry.
so might it be possible to gain some, I guess to me that suggests there's been selection specifically on shape size that's uncoupled it from the rest of the other tissues, and might be able to gain some insight into the function of it by looking at morphological or ecological correlates of, when species have an unusually high ratio, do they share other certain phenotypes or behaviors or ecologies compared to species that have at the other extreme that might suggest something about the sources of selection that are acting to uncouple this, and why scape length in particular would be important to vary. It suggests selection's favored variation in this trait independent, it's decoupled it.
Yeah, so that's a really good point, and something that Raji and I have talked about, but don't really know how to figure out. Because yes, maybe this is like a trait that has like geographical and ecologically I like distinct modules. So like maybe like a ratio that's a lot greater than one. So soldiers having larger escapes than workers is maybe in like one region or another. It could be coupled with other traits, like you said. We've been thinking about like head width, or like not head width, sorry, but just like head shape in general, and many other things. But it's definitely a good point. I
Yeah, I
also
guess the
just
problem is it's kind of a fishing trip that
yeah,
you might luckily hit upon a
yeah,
behavior or an ecological trait or a morphological trait that correlates strongly with that, but you may not as well. It could be
exactly.
yeah.
So it's kind
Anyways,
of just guessing. But for now, I guess we can more easily do the morphological side of things. So like because those pictures are available, we can like see if it correlates with some other trait on the head or its body. And like that textbook also had like geographical information. So like if it was in Brazil or like South America. So there's definitely possibilities. And I guess I just need to learn how to code. Yeah.
Interesting. If there are any other questions, you can feel free to use the chat or the text or turn on your mic. Excuse me.
Yeah, one of my questions was for the Phi Doli outliers, or just general for that, for where there were outliers that you might expect, where the soldier's larger, and outliers that you wouldn't expect where the worker's larger.
Yes.
Was there any phylogenetic signal to that? Were those that had the different ratios in the same clades within Phi Doli? Phi Doli?
So I actually didn't check that, but that's a really good point. Um, there are more intensive like phytologenies out there, and so I could see if there's a correlation between those extremities and areas on that phylogeny for sure. Um, yeah.
Yeah, just to know if it like decouple, like are each one of those outliers in one extreme, are each one of those outliers a an independent decoupling of the trait, or is it one decoupling that then gave rise to all those outliers?
Absolutely. So I think that definitely falls in this next step category because I do want to look at the
Hmm.
Phytoli genus in more depth now that I've kind of gone in like a bigger idea of what's happening. And so in addition to, I guess, like finding allometric patterns across this um, like dimorphic escape ratio, I could see if there's signatures of um like a specific node or something where like
Hmm.
all of these species have a DSR greater than 1 or something like that, or DSR of 1 if there's some node there for sure. So I guess just future work to be done. It never ends really.
Yeah,
Um,
1B or something for the slide.
exactly. For sure.
Any other questions in the audience?
I have one more question,
Sure.
and then I'll stop.
No,
Uh,
keep going if you want to.
no, it was just so there's a lot of variation within a certain gene within, like for example, Phi between the ratio of worker to soldier. Um, and then when you were looking at when you had the phylogeny up from the different genera all across ants when you went to Cornell,
Yes.
um, but you look at and maybe I'm wrong on this. You look at one species in a given genus, or like a handful of species in a given genus, and then conclude where the decoupling arose. But could you have just got lucky or unlucky, depending on how you want to look at it, where you selected one species that like did have it decoupled, but then you like didn't happen to sample species in the same genus that still had it coupled? And so then
Yes.
your inference of the node is like depends on that.
Oh, it's clear for sure.
Yeah.
So I'll just go back to the image here. So my time at Cornell unfortunately was limited. So I was only able to spend a week, and the microscope there also had other people using it. So we really tried to focus on getting like I guess a larger sample across the ant phylogeny.
Mm.
And even when it came to Cephalotes varians, like their antenna, let me see if I can find the picture, are like tucked inside their heads. So I literally couldn't measure them. But very luckily, someone in the Moreau lab actually had just dead samples in a file. And so I was able to use their their setup to dissect it actually to remove the antenna and measure it. Um, and so yes, it could have been lucky. The same way that I got lucky with Phydola dentata, because if we had a different Phydola species that
Right.
had a DSR greater than
Hmm.
1, I would have never even noticed, right? Or none of us would
Hmm.
have really. Um, and so I guess like something that could happen is like I do this for cephalotes to see if it's across the cephalotes uh genus or if it's just like in this one Cephalotes variant species. So yeah, definitely it might have definitely been lucky for sure. But yes, I guess I don't have enough information to really drive home this point until I get a couple other data points.
Yeah, I think you could say it like it could be decoupled there, but it could be further back. Yeah,
Yes,
or it could be multiple
for sure.
decouplings within
Um,
the, yeah. Yeah, I
so
got
it's
you.
kind of like
Mhm.
I guess the first instance of this idea of even traits being decoupled from body size. So there's still a lot more work to be done, a lot more traits to be looked at, because this is just escape, right? Are there other traits across the ants that are decoupled, like I don't know, something on their leg or something? So there's definitely more to be looked at for sure.
Yeah, great. Great. Yeah, great talk. I'm sure I speak for the whole audience. We enjoyed seeing your passion for the ant measurements and the ants.
Thank you.
So I really appreciate it.
Yeah, thank you very much.
Awesome. Thank you so much, everyone, for attending and for questions and just showing up. And if you have any questions, feel free to email me. I'm more than happy to answer anything.
All right, great. So I think we'll end it there. And thanks so much, Erica. And we will see you guys next week for next week's Greg seminar. See you guys.
Okay.